So what we have been doing for some time in a very broad overview with a lot of emphasis on concepts is to look at the central dogma, which is what we started with. And while doing the central dogma, I have introduced you to proteins and DNA and RNA as macromolecules, as major biomolecules. And you've also seen some of the machinery in the cell and how information is transferred from the storage molecule to the two executive molecules. And that has been a big chunk of what I've been discussing with you. In the last few lectures, I've concentrated a little bit more on proteins, though I won't give it too much time. We'll also touch upon lipids and also touch upon carbohydrates. Okay. All right. So the picture over here is the broad area which I'll be covering. Central dogma, you know. This is again a ribbon representation of a protein, very large complex inside the cell called the proteasome, and there are hundreds and thousands of interesting proteins. This is interesting because it's a recycling system. It basically takes up proteins which are old and which need to be recycled, takes it inside. There's a pore inside which is not obvious. Think of it as a thermos flask with a hole at both ends. The protein goes in through the hole in the top, gets chopped up into little pieces, and gets chucked out from the other side, the pore on the other side. Here are lipid fat droplets inside a cell. And one of the things which I hope I have conveyed to you through the various animations is that the environment inside the cell is very crowded with these macro and micromolecules. It's not like a beaker of water with a little bit of sugar in it. It's an extremely crowded environment. And what you see here in yellow is fat, basically lipids, which we are going to talk about today. And in blue are mostly proteins. And of course, there are water molecules pretty much everywhere. And on the right is the idea that carbohydrates are an energy source. And this talks about the basic conversions between the major carbohydrates like glucose, fructose, galactose inside the cell, which again is part of metabolism. So the linear sequence of amino acids, when it comes out of the ribosome, is not in a folded state. It is in what we call as an unfolded state. It has an N terminus and it has a C terminus. Just like nucleic acids have a 5' 3', there is polarity in the polypeptide chain with the N and the C terminus. All right. So let's assume synthesis starts from the N terminus side, and let's say this much part of the protein has been synthesized while the rest is being synthesized. This part, as it comes out, starts to fold. Okay. And this is happening inside the cell. In vitro, if you take an unfolded state and put it in solution, it will start to fold. But it will start to fold as a single large entity. Now, each region of this polypeptide chain will try and form secondary structure. And the two major secondary structures, as you are now aware of, are beta sheets and alpha helices. Okay. And this particular example is of a protein which is just alpha helices and turns, nothing else. There are no beta sheets over here. So this protein starts to show local folding, and there will be different steps in this process of this protein folding locally, then the local folds, that is the alpha helices coming together to form the folded state. And this is the so-called folded state, which is the functional state of this particular protein. Now, as the molecule transits from the unfolded state to the folded state, and this is a very fast process, happens in microseconds, let's say. There's a huge range. It's from nanoseconds to milliseconds, depending on the protein. The larger the protein, the slower it is to fold. But broadly, it's a very fast process. And it is exploring a lot of possibilities as it keeps on folding. And these possibilities include maybe transiently during the intermediate states. Some of the chains tend to form a transient, let's say, beta sheet. But finally, the protein will not show a beta sheet. Also, in the path for forming the native or the unique native state, there will be many, many different ways by which, it's like a Rubik's Cube. There'll be many permutations and combinations by which you will attain the final state. And you have to explore all of these states as you go from the U state to the N state. Fine. Now, when biophysicists study protein folding, they simplify things and say much of the time there are only two states present, because once a protein folds, it's mostly in the folded state, and the spontaneity of folding is driven by a negative delta G, which is basically in the range of 5 to 15 kilocals per mole. Okay. And when we did a calculation, we basically found that if you, for a protein which has a delta G of minus 5 to 15 kilocal per mole, the the ratio when we calculate between the folded state and the unfolded state is 1 into 10 raised to 7, basically indicating that at any point in solution with a delta G of minus 10, 10 raised to 7 molecules are folded and one molecule is unfolded, and there is a equilibrium between these molecules. Okay, so this was a very fairly simple concept. Now all this seems very well and good, but I also told you that the bonds which are stabilizing the protein, and remember proteins are stabilized in water, these bonds are of variety of types. These include salt bridges, these include hydrogen bonds within the protein, especially secondary structure. They are van der Waals interactions, and there is also a packing in the core of the protein, and I'll try and draw it for you. At the core of the protein, many of the hydrophobic groups pack together away from water, and there is usually not much water inside the core of the protein. Water is completely excluded. Most of the water is either on the surface or in crevices of the protein. So all we know as of now, that experimentally we know that the proteins are stable to the tune of 5 to 10 kilocals per mole. We know that hydrogen bonds have a strength of around, let's say, 3 to 4 kilocals per mole. We know breaking of a covalent bond is about 80 kilocals per mole. So in theory, breaking of a single covalent bond can destabilize the folded state. Breaking of two hydrogen bonds can severely destabilize the folded state. In spite of the fact, any mutation can also destabilize the folded state. So in spite of that, proteins are folded, they are functional. All right. Now, I'll wait for questions before I go ahead. Is there anything over here which you didn't understand?
So could you move to the previous slides where the folded, unfolded state of protein was there?
Yeah.
This one.
Yeah.
I see that there's an equilibrium between U state and first state,
Yes.
and then the N state. What does the partially folded protein do in our body? Like, what is the purpose of it? Because it would be unstable.
Right. So partially folded states. So let me tell you the traditional viewpoint. Okay. The traditional viewpoint is the viewpoint from about 10, 15 years ago. The traditional viewpoint is that proteins are made as unfolded states and they need to function as folded states. And the only way to go to the native state is to go through partially folded states. So partially folded states do not need to have any function. They are transiently present immediately after the protein comes off the ribosome. So that is the classical view. Fine. And
very short lived.
it's very, so remember the entire process of folding is in microseconds. So
Okay.
Intermediate state, since there will be maybe 1,000 intermediate states during the folding of a protein of different types, each intermediate state will probably be there for a picosecond. Okay.
And so one more thing from the previous lecture, could I ask?
Yeah, please.
So while you were explaining about a Ramachandran map, you explained while drawing a sequence of protein in which you took different amino acids. And then you told that I do understand that we plot the coordinates of the phi, psi, and omega and in the Ramachandran map. But how can we infer the configuration or where the density of that particular amino acid increases if we are taking a protein which consists of different amino acid? I mean, it should be of same amino acid, and then we should try the permutations and then we can plot.
So let me try and give you a short explanation. I think there is a disconnect over here. So this particular unfolded state over here, which you can see drawn over here, you can see that, right? This is basically a sequence of amino acids. Fine. Now, all I'm saying is that if each block over here, now I'll just use some other color. If each block over here represents one amino acid, okay? You agree with that?
One same amino acid.
No, it's not a same amino acid. It's different amino acids. Okay. If each block represents an amino acid and there are 100 of them in this unfolded state, all I'm saying is for each block, fine, which contains a phi, now that's better, psi, and omega. And we ignore the omega because it's always 180 degrees. For each block, we have a phi and we have a psi value. And in the Ramachandran map, we just plot for each block. X-axis is one, y-axis is another. We just plot a dot. So that's the Ramachandran map. And the next amino acid is a completely different amino acid, but the phi psi are main chain torsion angles. They have really nothing to do with the side chain. Well, indirectly they have something to do with the side chain, but the atom used for plotting phi and psi are all main chain atoms. What the R group is not used for the calculation at all.
So that gives the tells about the protein structure, not how many configuration that particular.
No. So the Ramachandran map is only a representation of the native state, because that is the state.
Okay, I got it.
Okay, that's the state represented by X-ray crystallography. During folding, the protein will follow basic rules of chemistry, but it may have a more broader representation in Ramachandran space than the folded state.
Yes, sir. Yes, sir.
Great.
Thank you. Yes, sir.
Okay. Are there any questions?
A question about the Ramachandran map. Should I
Yeah.
ask it now? The map for individual amino acids, right?
Yes.
For glycine and proline. So over there, the glycine one, for example, represents all of the phi psi angles glycine can take, with some being more favorable than others.
Right.
But then, and you also showed us a more generic map which had all of the amino acids of all proteins plotted.
Right.
So, how is it that the glycine map covers more area than the generic map? Because everything that is plotted on the glycine map should also be on the map with all other amino acids, right?
Okay, so let's see if I think here again there is a different way of looking at things. This particular, let me take this unfolded state again, okay, or the folded state over here. Let's talk about the folded state. The folded state over here, this molecule, let's assume that it is made up of 100 amino acids, okay. There will be not more than, let us say, 5 glycines in this linear sequence. So if you draw a Ramachandra map for this particular protein, you will have 99 dots and only 5 of them will be glycine. And those 5 glycines can be here, can be here, can be here, or can be here in the Ramachandra map.
Okay.
So if you look at dots in the
Right.
Ramachandran map, only one will be here, the others will be in Ramachandran, normal Ramachandran space. Is that clear?
Yes, so but the map you showed, I think for all of the amino acids didn't have any dots at all in the fourth quadrant.
So that's, see, glycine is free to explore more areas of conformational space than any other amino acid. Just because it's free to explore doesn't mean it always explores, because it is also part of a main chain surrounded by tryptophans, tyrosines, bulky amino acids.
Okay, yeah, understood.
Can and will are two different things over here.
Okay, so yeah, understood.
A question?
Sir?
Yeah, go ahead.
Sir, I have a doubt. What is this 5 to 10 kilocalorie energy? It is delta G of protein, or it is the energy of peptide bond?
So the 5 to 10 kilocals per mole is a representation of the stability of the folded state over the unfolded state, and it's basically the overall stability of the folded state with respect to the unfolded state. And you can relate the delta G through two main equations in biochemistry. One is a relationship between delta G with the equilibrium between the two states, and one is the relationship between delta G with enthalpy and entropy. Not clear enough even now?
No, sir. Okay.
Okay, so
Sir,
yeah.
whatever map we drew was for one particular protein. Like when we talk about glycine and we do the Ramachandran map, it was for polyglycine.
Yes,
For proline, it was for
a
polyproline.
polyglycine map will show you dots in all 4 quadrants.
That is fine. But we never drew a generic graph for all the amino acid of all the proteins that exist, right?
Yes, we did.
For all the proteins.
Pretty much, means all the proteins, means whoever drew it at whatever point drew it for pretty much all the proteins. For example, if you look at this one over here, if you look at the number of dots on the picture, there are probably 100,000 dots over here, right? Many, many dots. Look at all the little dots behind. These are not shadings, these are dots. So this is a representation of many proteins. This is probably a representation of about 10 or 12 proteins, or a very one large single protein. Is that clear? Okay. I need you to conceptually understand that the ribosome makes a polypeptide. It goes through intermediate forms. It forms a native state. The native state is the functional state. And we have ways to measure how stable a protein is. And surprisingly, the protein is stable moderately, because 5 to 10 kilocals per mole, which is the stability of this protein over here. If I break one hydrogen bond, the stability goes from 10 kilocals per mole to 8 kilocals per mole. I break two hydrogen bonds, it goes from 8 kilocals per mole to 6 kilocals per mole. So I make one mutation, it goes from a stability of 10 kilocals per mole to about, let's say, 3 kilocals per mole. So this is stable and but weakly stable. And the point I was trying to make is that when you look at the different forces, so let's not go into the thermodynamics of it for this course. So this slide is basically not part of the course. It is a balance of different forces, which are enthalpic and entropic, okay, which we can discuss tomorrow morning. And when you add up all the forces which are involved in stabilizing and destabilizing a protein, the net stability is, as is written over here, is very small. Okay, so this is the number which is used, whereas the forces which are against and for are very large forces. Fine. With the fact that an unfolded state with a large entropy folds into a folded state with a very with a very low entropy. There's an increase in order over here, opposes uh, the spontaneous free energy folding, simply because the entropic term over here, it has to, if it goes up, there is more disorder. If it goes down, there is less disorder. And the folding of a chain basically lowers entropy. And if it lowers entropy, it's a negative term. It's a negative term. The negative of a negative is positive. And because this term becomes positive, delta G becomes positive. And for a spontaneous folding, we want the delta G to be negative. Okay. So all this is really not part of the course, but I'm just trying to give you a quick explanation. Fine. Okay. So I just want to leave you with this idea of the variety of proteins, large proteins, small proteins, many of them doing functional roles, and the broad ideas about protein stability, the fact that there are multiple stabilizing forces involved, everything is in water, and there is something called as a protein folding problem, which has existed for over 60-80 years, and there are different aspects of the protein folding problem. One is the time aspect. How is a protein folding so quickly? Because one assumes it has to explore a lot of conformational space to find its final native state, right? It's like a Rubik's cube again. You start with a Rubik's cube which has been shuffled, and if you have not done Rubik's cube before, you can spend hours and hours trying different combinations, trying to solve the Rubik's cube. And if each movement of your hands takes you, let us say, a tenth of a second, and it takes you 10 seconds to make 10 moves, and you keep on doing these moves and you never find the solution, that's what can happen to a protein. Okay. Second thing I've been telling you is there is a relationship between the sequence of amino acids. These are not random. Each folded state is in its folded state because of a unique sequence which is there. So hemoglobin in all species, starting from hemoglobin like entities in, let us say, invertebrates, and myoglobin, they all have very conserved sequence of amino acids. And this conservation is very important, because if you lose this conservation of the linear sequence of amino acids, you will not get the folded state. And the third part of the protein folding problem is this whole idea that proteins are weakly stable. Any small push on the wrong side can force them into going to the unfolded state. And going to the unfolded state is a disaster, because the unfolded state is not a functional state. For hemoglobin to work, it has to be in its folded state. In the unfolded state, it will not be able to bind oxygen. So folding is a very, very critical physical chemistry phenomena, biological phenomena, physics phenomena in going on inside the cell. And every second, hundreds and thousands of proteins in your body are being made by the ribosome and are folding. And if they don't fold properly, basically life doesn't exist.
So
Yeah.
sorry to worry you again about the Ramachandran map, but one thing isn't clear now also. So, for example, I'm taking a lysine amino acid, and I want to know in how many configurations that particular amino acid can exist, in terms of how many angles of phi, psi, and omega is same 180 degree it can undergo, like naturally in our body.
Right.
So for that, for plotting that particular thing, I need to know one particular protein in which all that possible configuration should exist. Am I right?
Not really, you see.
So then how did we got the idea about, like, we have all the maps of amino acid, all the amino acid in which what all configuration they can exist. So how can we get the idea if we are taking a protein in which there are different amino acids? Then how can we plot and be very sure about that, okay, this particular amino acid cannot go into that phi psi angle, and only this one, like we
So
plotted.
nobody's saying an amino acid, well, people are saying an amino acid cannot go to a certain phi psi configuration. The generic statement is except glycine and proline, all other amino acids can only go into 30% of the conformational space available. And the reason they can only go into the 30% of the conformational space available is because they are in a linear sequence, one after the other. If you rotate around a torsion angle, the R groups bang against each other. And because they bang against each other, some areas of torsional space which are allowed to glycine are not allowed to any amino acid, alanine onwards. Alanine to tryptophan, no amino acid can go into certain areas of conformational space.
So scientists have plotted all the proteins, and then they have figured out.
Exactly.
Okay. Now I got it.
So
Thank you.
Ramachandran did this without having structures. He used models. So he predicted this without even having the data we have today. But every year when another 1,000 structures are solved, we know Ramachandran was perfectly correct. Every structure validates his initial modeling.
Okay, sir.
Okay, fine. All right. So,
sir, here D means, D, that D to N part, D means destabilized, na?
Yes, I'm sorry, different nomenclature is used. D is the denatured state or the unfolded state.
Okay, sir, denatured. Okay.
And N is the native state or the folded state.